Ms. Katie for concluding this session. We have already very good discussions and talks so far, very insightful talks. And now to move further, we have another speaker with us, Dr. Arun Kumar. And I feel privileged to introduce Dr. Arun Kumar to all of you. Dr. Arun Kumar is presently the principal scientist in the fermentation and phytofarming division at CSIR IHPT Palampur. Dr. Kumar is an accomplished scientist with extensive expertise in enzymology, enzyme engineering, metabolic engineering, functional genomics, and plant biotechnology. Over the course of his distinguished career, he has served at renowned institutions including the University of Wisconsin, Madison, USA, McGill University, Canada, and Punjab Agricultural University, Ludhiana. His research has significantly advanced understanding of industrial enzymes, crop improvement, genome editing, plant stress biology, and sustainable biotechnological solutions for agriculture and industry. Dr. Kumar has led several nationally funded research projects and published about 50 research articles in high-impact international journals, has secured 4 patents, and successfully transferred innovative technologies to the industries. He has been awarded several prestigious awards from SRB DST, travel awards from USA and from DST and INSA. His contributions have earned him recognition both nationally and internationally, and he continues to play a leading role in advancing scientific research and innovations. So this is a time to move from the stresses, from the disasters to the world of microbes, and he's going to talk about innovations in the enzyme technologies and plant microbe innovations for a sustainable future. So please, Dr. Kumar, come up on the dais and share your knowledge.
Namaskar, a very good afternoon to all of you. Respected principal sir, distinguished speakers of the day, faculty members, and dear students. So, I would like to thank Dr. Arun Kumar for inviting me to this very event and introducing me. So, moving on to the presentation. So, title of my today's talk is Innovations in Enzyme Technologies and Plant Microbe Interaction for a Sustainable Future. So before I move to my full presentation, I would like to give a brief overview of, is it okay if I use this mic? About our institute before I move to actual presentation. So many of you might be aware of this institution. It is one of the constituent laboratories of CSIR. It's named as CSIR Institute of Marine Biotechnology. This institute was established in 1983 with a vision to become a global leader on technologies for boosting bioeconomy through sustainable utilization of Himalayan bioresources. And the mission of our institute is to discover, innovate, develop and disseminate technologies, processes, products and knowledge from Himalayan bioresources for boosting the bioeconomy of the region. We work on all kinds of bioresources, including plants, animals, fungi, bacteria, insects, viruses, etc. And we have 6 divisions in our institute: agrotechnology, biotechnology, chemical technology, dietetics and nutrition, environmental science, and fermentation, phytofarming. And we work on a wide range of crops, for example, industrial crops, floriculture, horticulture, medicinal and aromatic plants, industrial enzymes. So our group is mainly working on industrial enzymes. This is another center of CSIR IHPT, which is called Center for High Altitude Plant Biology. It is located at Kelong, Lowland Spiti district of Himachal Pradesh. So this center was established with the aim to study biosystems at high altitude, vis-a-vis climate change, to study the ecology and plant adaptation behavior at high altitudes, and bioprospection of bioresources, ex situ conservation,
of indigenous flora over there and cater to the needs of tribal communities for inclusive growth. So that is a brief introduction about our institute. Now, coming on to the actual topic of today's talk, this is a brief flow of my presentation. I will be giving a brief introduction about enzymes, why enzymes are important, and what is the commercial importance of enzymes. After that, I will walk you through what is required to harness these enzymes for industrial use, and then an overview of enzyme research related activities that are being carried out in our institute. And I will follow up this with 2 case studies. First one will be on superoxide dismutase enzyme that we have discovered from a plant biosource. And another one will be EPSP synthase, which will be in respect to plant pathogen interactions. So, if I ask you one question, why studying enzymes is important? So, probably we have biology students here and they might be able to answer it. But for others from other backgrounds, they might also be aware that enzymes are important because they catalyze different kind of biological reactions. So, being a living object, there are a lot of metabolic activities that are happening inside you. Same is true for plants also. So, enzymes act as catalysts. Basically, what do? They enhance the rate of a reaction by lowering the activation energy of reaction. So basically they are important if you want to live. If one of the enzymes of your metabolic process ceases, you won't be able to survive. You will be diseased. So this is a brief definition of enzymes. Enzymes are typically defined as biological catalysts, usually proteins that speed up chemical reactions within living organisms. They enhance the rate of reaction by lowering the activation energy, and they are very selective in nature. Okay, so this is just a brief introduction about enzymes. But why enzymes are important and what is the industrial perspective of enzymes? So if you look at the industrial perspective of enzymes, they have significance in different walks of life. They have clinical importance. For example, if you go to hospital, you get sick, and doctor asks you, clinician asks you to go for certain tests. So, many of you are aware that there are certain tests like if you have a liver disease, doctor will ask you to go for, measurement of SGPT, SGOT levels. So if your SGPT and SGOT levels are high, that means you have a fatty liver. So likewise, there are certain enzymes like creatine phosphokinase. So they tell you about your heart is functioning properly or not. Likewise, there is prostatic acid phosphate. So these enzymes, they act as biomarkers. Another importance of enzymes is drug development. Every medicine that you take, they have one of the enzymes as their target. If we talk about penicillin, which you take when you get infected, penicillin acts by inhibiting a transpeptidase of bacteria. So thereby, it doesn't allow bacteria to grow. So this is another example. And I have picked up a few examples just to show that enzymes are really important in day-to-day work. Enzymes are also important in agriculture sector. You talk about any herbicide or pesticide that is being used, they are also targeting enzymes. For example, you might be aware of a pesticide, herbicide, glyphosate. So what does this glyphosate do? It basically inhibits EPSP synthase, which is one of the essential, not essential, but it's a rate limiting step of shikimate pathway that is operational inside the living organisms. So basically this pathway is responsible for production of aromatic amino acids. So basically glyphosate targets that EPSP synthase. So what happens, once you disrupt this pathway, the plant is not able to survive. So it is based on that principle. So this is a brief introduction about enzymes and their industrial importance. So now, if you want to be a global leader in enzyme production, you have to, be self-reliant in terms of their production. So, how can we achieve that? You see, India is currently, producing enzymes, and the market is estimated to be around 3,000 crores, and is expected to increase further. But if you look at the global enzyme market, it is around $14 billion. So this is a huge gap. If you want to reach to that level, you need to have your indigenous technologies in terms of discovery of enzymes, innovations around enzymes and their production. So, if you want to do this, what is needed? So, you need to have a basic understanding of enzymes, what kind of catalytic reactions they catalyze, what kind of cofactors they use there for proper catalysis, what is their substrate specificity, what is their pH range, what is their thermal stability. So you need to have proper profiling of enzymes for all biochemical parameters. And once you have that, you need to have cost-effective production systems. And if you want to use them for human applications, you need to have proper doses, bioavailability studies, and safety assessment studies. So these are some of the prerequisites that are required for if you want to be a leader in the entomology area. So with that brief introduction, I will tell you what facilities or what kind of capacity we have at CSIR ISPT. So we have all the state-of-the-art facilities in our institute, including all upscale production and purification facilities. We have bioreactors, cell separation systems, cell homogenizers, centrifuges, protein purification and lyophilization facilities. So this is just an overview of what kind of facilities we have. Now, what do we do in our lab? So all of my research is revolved around enzymes for different applications. So what we do is we rectify enzymes of plant resources or microbial resources. We engineer them for superior properties, and these are some of the areas where we target these enzymes for pharmaceutical and healthcare industry, biofuel industry, and agriculture industry. So due to paucity of time, I won't be able to cover all the aspects of what we are doing in our lab, but I'll just focus on two areas where I will tell you how did we identify one of the very important enzymes from a polyextremophile plant. It's the name of enzyme is superoxide dismutase, and another one is a story on EPSP synthase that is with regard to plant pathogen infections. So talking about superoxide dismutase, this story is from discovery to translational aspects. So I will tell you how this enzyme was discovered with some unique kinetic properties and what did we do. So story begins, I will come to the story later part, but you should be knowing why this enzyme is essential. Being aerobic organisms, be it plants or human beings, we need oxygen to carry out our daily metabolic activities. If you want to convert, you have food and you convert it into ATP, oxygen is essential for that. So that is the positive side of oxygen because it gives you energy. But at the same time, it is toxic also because there are certain byproducts of oxygen which are generated during routine metabolic activities such as superoxide anions, singlet oxygen, hydrogen peroxide, hydroxyl radicals. So these are collectively called as reactive oxygen species. So what they do is they actually react with all the biological macromolecules inside the cell. For example, they react with lipids, amino acids, proteins, nucleic acids, and ultimately what do they do is they kill the cell. There is membrane leakage and cell, it's kind of problem. So these are not very, these reactive oxygen species are very, very harmful to cells. So cell has to maintain a fine balance between the production of these reactive oxygen species and their scavenging, because at certain level they are important for certain signaling kind of things that are happening inside the cells. So they act as signaling molecules. But if they are produced in excess quantities, they will be harmful to the cells. So this superoxide dismutase is basically, it has been, known as the first line of defense because it catalyzes dismutation of superoxide anions into hydrogen peroxide, which is further taken care of by other enzymatic mechanisms which convert this H2O2 into water. So this protein was first isolated from animals as a green copper protein. It is also referred to as erythropoietin, endophenol oxidase, and tetrazoline oxidase. And there are different type of SODs that are being present inside the cells. So these are called isozymes. So basically, based on the metal cofactors that are being used by these superoxide dismutases, they are classified into 4 different classes: copper zinc SOD, Mn SOD, iron SOD, and nickel SOD. So basically, they are present in different compartments of the cell. So basically, these are present in different compartments of the cell, and they do site-specific dismutation of superoxide anions inside the cells. So if we talk about the commercial importance of these superoxide dismutases, they have wide range of applications ranging from pharmaceutical industry to cosmetic industry, food industry, and agriculture industry. And there are several products of SOD that are available in the market. If you look at the market potential of superoxide dismutase, it was evaluated as approximately $3.7 billion in 2021. And it is further expected to grow. And these are some of the key players in the superoxide dismutase industry. You will see that most of the market is dominated by USA, Europe, but we stand nowhere in terms of production of this enzyme. So our group, we started with some explorations in the high altitude regions of Lahore and Spiti. And this work was, initiated before I joined my PhD. So Dr. Sanjay Kumar's group, they were exploring these extreme niches of Himalayas, and they came across with a plant which is called as Potentillator sanguina. So you can see, this plant, it is growing under very harsh conditions. You see the partial pressure of gases is very low here, and there are a lot of temperature fluctuations. There is extremely cold environment over there. There is high intensity of light, heavy load of UV radiations. So this plant is kind of a polyextremophile. It is kind of surviving under very harsh conditions. You barely see any vegetation growing over there. So it was expected that this plant, why it is surviving under such harsh conditions, it potentially could have certain defense mechanisms which are allowing this plant to survive. And as I told, the superoxide dismutase is one of the important antioxidant enzymes. So it was anticipated that this plant could have that kind of SOD which is helping it to survive under harsh climatic conditions. So people started doing some experimentations here, and what they did is they brought this plant and exposed it to low temperatures, 4 degree and minus 20 degree centigrade. What they observed is that there was induction of this superoxide dismutase activity. The activity of this enzyme was, it went high when the plants were exposed to low temperatures. So we did more, studies on it, because there are different kind of isozymes. They observed 5 different kind of isozymes of SOD in the plant under controlled conditions. But what they observed is, when they exposed this plant to low temperature stress, there was induction of one isozyme which is labeled as isozyme C here, and also one fuzzy band around the one which is labeled as F. So there is a lot of inhibitor studies, including suppression of this chloroplastic and mitochondrial this protein synthesis inhibitors, and also they inhibited the nuclear synthesis of nuclear proteins using different inhibitors. So what they observed is that this isozyme C, which is present here, it is a copper zinc SOD. So further, this protein was purified, and they also looked at whether this protein is active at low temperature only, or is it able to tolerate high temperatures also. So what they observed is, even if you heat this enzyme to extreme temperatures, including autoclaving, this enzyme retains activity to a great extent. So this enzyme was further isolated, crystallized, and this is the crystal structure of this enzyme. So this is basically this enzyme, it is a homodimeric enzyme. So there are two subunits of this copper zinc SOD, and on the right side it is a catalytic site where copper ion, which is represented as W here, it is interacting with other amino acids which are important for oxidation reduction reactions. So this was the crystal structure of this enzyme. So what was unique about this enzyme is that when we did comparative analysis across all the copper zinc SODs reported in the literature, we observed that this enzyme has gap volume which was lowest among all the reported SODs. The gap volume is the gap between two subunits. It was lowest, so it was so compact. Probably that was the reason it was so robust kind of enzyme. It was resisting heat-mediated denaturation. So we further asked, can we engineer this enzyme to further enhance its kinetic properties, including thermal stability or catalytic activity? So based on the crystal structure, we identified few amino acid residues which might be important for its activity. So these are some of the listed amino acid residues which were identified and mutated into alanine. So what we see here, when we mutated these amino acid into alanine, there was one mutation which resulted in enhanced activity even after autoclaving the enzyme. This mutant was labeled as C95A. There were certain residues where activity was completely abolished. So out of all these mutations, we just focused on three mutations. One was around C95, C56, and 145A. So when we looked at the crystal structure of this protein, what we observed that these 1645, 56, and 16145, they make inter-subunit and intra-subunit disulfide linkages, and probably they are important for maintaining the structural integrity of this enzyme. Whereas the 1695, which is shown as a yellow dotted sphere in the middle, this 1695 is present at the dimer interface. And this is a bulky amino acid. So if you convert into alanine, what happens, there is increase in the monomerization of the enzyme. This experiment just suggests that once you remove this cysteine residue, the enzyme is converted from a dimeric form into monomeric form, which is more stable. So these were different experiments just to prove that this enzyme is being converted from a dimeric form to monomeric form. So this is a size exclusion chromatography experiment. And again, the protein was purified and just shown that this 1695A mutant is superior in nature compared to the wild type mutant. These are other biochemical properties where we show that this mutant, it is outperforming wild type enzyme and has a temperature optima that is different from wild type enzyme. This is an interesting fact. If you look at this graph, it shows that the wild type enzyme has half-life of thermal inactivation of around 1 minute, and this C95A mutant, it is almost double, 345 minutes. This is another study that we did to assess whether this enzyme can tolerate pyrolysis, related pyrolysis. We subjected this enzyme to various enzymes like trypsin, chymotrypsin, and papain, because we want to see if we give oral application of this enzyme to human subjects, whether it can pass through the gut without being damaged. So these were some of the important experiments that were important to assess whether it can tolerate proteolytic cleavage by these. So as you can see, there was little bit shift in the of the bands, but it was more or less quite stable to limited proteolysis. This was also confirmed by SD Spade, and few more studies related to CD spectroscopy, beyond your scope, but overall what we proved here is that this enzyme is quite thermostable, and this was the first report we reported that enzyme can be autoclaved. Even after autoclaving, this enzyme retains its activity. This enzyme is functional at 0 and sub-zero temperatures, which was again a first report from our institute. And so once we did all this, we published this work also, filed patents also on this. The next thing was, if you want to use this enzyme for human applications, you need to have all the safety assessments of this enzyme. So we did a lot of safety assessment test on this enzyme. We collaborated with CSIR, CCMB, Hyderabad, and a lot of tests were just done to ascertain that this enzyme is safe for human applications. We also did certain experiments on mouse models, where we did subcutaneous injection of this enzyme at a very high dose, approximately 2,000 units per kg of body weight. And you see there were no lesions in the liver and kidney, suggesting that this enzyme is quite safe for intravenous applications also. We did some experiments on zebrafish models also. You see here, these are different developmental stages of zebrafish embryos, which were treated with very high dose of this copper-nic acid. You see, there were no defects in the development of this zebrafish. So once we established that, this enzyme is safe for use, from industrial perspective, we wanted to have production process for this enzyme at a very high scale. So we optimized different fermentation parameters to achieve high cell density, because high cell density would lead to production of more enzyme. So we reduced the normally it was taking 23 hours to grow the cells to very high scale, but we did certain permutation combinations and reduced this fermentation cycle to 12 hours. And we could achieve a yield of approximately 300 milligrams of protein per liter. These are the gel pictures. So once we did all these things, next thing was technology transfer, because we have a technology ready to transfer to industry. So we transferred this technology for production of SOD to an industry, named as Biotech Private Limited. So they had exclusive rights on this technology for its production and sale for different applications. So this was one story related to copper-nic SOD, but as I mentioned, there are other isozymes of copper-nic SOD also. For example, MnSOD, nickel SOD, iron SOD. So this is another story. I'll quickly cover it out. This is related to induction of a new isozyme of SOD, which is named as mitochondrial superoxide dismutase. It is present in mitochondria of plants. So basically, this enzyme was found in tea, Camellia sinensis. One of the clones, tea is during winters it undergoes dormancy due to prolonged winter spells. So one of the clones called Tinali clone, which is grown in our experimental farm, it has a very short period of dormancy, and this short period of winter dormancy was physiologically linked to the induction of this new isozyme. So what we did is we cloned this gene from Tinali clone. It is existing as a homotetramer. There are basically 4 different subunits of this enzyme. Because the crystal structure of this enzyme was not known, we did the in silico predictions. The only crystal structure that was there in the literature was of Arabidopsis, which is a model plant. So it is aligning perfectly with the Arabidopsis MnSOD. And we also showed with using different techniques that this is a homotetrameric enzyme, and it has a tendency to oligomerize. The major form of this protein is tetramer, and there are other oligomeric forms also. So, like characterization of copper-nic SOD, we also did biochemical evaluations about the activity and stability of this enzyme. So this enzyme, it works from a pH range of 5 to 10. It also has a temperature optimum of 4 degrees centigrade. And its KMB max, these are some of the parameters that are important, for substrate specificity, affinity of the enzyme towards their substrate. So this is regarding its thermal stability. In the gel picture, you see that this enzyme is quite stable after heating at different temperatures until 70 degrees centigrade. But once you move to 80 degrees centigrade, it is up approximately 50% reduction in the enzyme activity, and at 90 degree complete loss of activity. So we calculated its half-life of thermal inactivation at 70 degrees centigrade, and it was found to be around 176 minutes after heating at 70 degrees centigrade. So it is quite thermostable enzyme, and it has a melting temperature of around 80 degrees centigrade, which is correlating well with the in the first two studies, this melting temperature was determined using differential scanning calorimeter. And again, these observations were also validated with some other studies, seed spectroscopic studies also, which also, tells us that this enzyme is quite stable. It has the melting temperatures was determined to be approximately 80 degrees centigrade, which is corroborating well with the DSC studies. And we implicated here that the alpha helical regions of this protein are important for maintaining thermal stability, as there was sharp decline in the alpha helical content as we heated the protein from 70 degree to 80 degree to 90 degrees centigrade. So this was, another work on SOD that still is continuing. We are looking at different application aspects of this mitochondrial superoxide dismutase, how this mitochondrial superoxide is important for, plant abiotic stress tolerance, and also in animal studies we are loading this MNSOD on polylactic glycolic acid to form certain nanoparticles to manage ischemic stroke, where mitochondrial dysfunction is quite obvious. So this protein again it is also very tolerant to different inhibitors. This is from industrial perspective. When you use enzymes in different applications, they should be quite resistant to different inhibitors. So we studied the effect of different inhibitors, chelators, denaturantes, and this enzyme was found to be quite stable, except for DDT, where there is a sharp decline. So if we did some comparative studies here, and our chemistry sciences SOD is on the top. You see, this is there are other thermostable MNSODs reported in the literature, but this MNSOD from chemistry sciences, it is one of the most thermostable plant-derived MNSOD reported so far from our lab. So work is still continuing this aspect. So I conclude here on MNSOD. Now I will come on to another topic, which is understanding the molecular intricacies of rice-arsenine interactions to the sheath blight disease in rice. So this project, I was funded through the Ramanjan Fellowship when I switched from US to PAU, Punjab Agricultural University, Ludhiana. So I got this project for 5 years. So although there are several different angles to this work, but I will, because my topic is related to enzymes, so I will converge my talk, just pointing to the role of enzymes in plant pathogen interactions. So brief introduction about rice. We all eat rice, and we know that rice is also affected by several biotic and abiotic stresses. And there are several diseases like several diseases that affect the rice production, like bacterial blight disease is there, bacterial blight disease, sheath blight disease. So this disease, sheath blight disease, it has been rated as one of the most devastating diseases recently, and it has been rated as number 1 disease in terms of crop losses. There was one study published in Nature where they rated this sheath blight disease as one of the most devastating diseases. So this disease is basically caused by a soil-borne necrotrophic fungal pathogen, so which is present in the soil itself, and when there are conditions which are congenial for its germination, it forms hypha, it recognizes the plant and it starts penetrating the cells. So people have tried to dissect the resistance mechanisms against this pathogen, but breeders, pathologists, they eventually failed because there is no natural source of resistance against this disease. People have evaluated wide range of germplasm, but they couldn't identify a good level of resistance in plant, because breeders, they normally rely on resistant sources. What do? They identify resistant sources and try to transfer the traits to susceptible ones. But here in this case, because there is no absolute level of resistance, the plants, they just vary in the level of tolerance. So people identified some QTLs which are called as quantitative trait loci. So these QTLs are nothing but they are certain signatures, they are regions in the genome that are linked to that particular trait. But these QTLs were minor in nature. They were explaining very low phenotypic variation. So what we hypothesize is that if we are able to understand rice-arsenine interactions at a molecular level, it can help us devise certain strategies to engineer rice for tolerance to this particular disease. So there are two possible ways to address this thing. One is either you identify host susceptibility factors, how pathogen manages to manipulate host defense response. So there would be certain genes in the host which are being manipulated by the pathogen for its survival and successful invasion inside the plant. So if you are able to identify those susceptibility genes, you can edit them or you can knock them out. The other possibility is you identify the genes which are essential for pathogen virulence. And if you can block those genes, this is another way of developing resistance. So with that, assumptions and hypotheses, we selected two different rice genotypes. The one is called SHBR, which is moderately tolerant to Shigella disease, and another one was PRS, which is susceptible to this disease. So we did a lot of biochemical studies, histological studies, microscopic studies to discriminate the level of resistance among these two genotypes. You see, on the left side, right side, there is one graph showing this MDE content, which represents the electrolyte leakage inside the cell. So once this pathogen infects, the susceptible genotype has higher MDE content, and also the fungal biomass, accumulation of this fungal pathogen inside the susceptible plant is very high. And if you look at these microscopic pictures here, where we did a staining which is specific to fungal cell wall, this is called FIDC staining. You see that in susceptible versus resistant plants, you see there is more accumulation of hypha in the susceptible genotype compared to the tolerant genotype. So these were some of the basic things that we did just to discriminate the level of tolerance against this pathogen, and also to identify the stages which would be best to do further studies. So basically, based on these studies, we divided the stages of infection of this pathogen to initial recognition stage, which was 24 hours after infection, a disease establishment stage at 48 hours, where this fungal hypha they were well established inside the plant, and then necrotrophic phase at 72 hours, where plant this fungus it was growing like anything. It was killing the cells because it's a necrotrophic pathogen. It has to kill the cells to feed upon the dead tissue. So what we did is, there is a technique called RNA sequencing, where what you can do is, you can sequence all the expressed part of the genome in a plant. So this is a technique that is being routinely used in molecular biology labs. So what we did is, we collected RNA, we infected the rice cells, and we collected RNA at these three different stages. And we did a global transcriptome analysis, where we could capture all the transcripts that were being expressed, and then map those transcripts to rice genome. So most of the reads, because it was a pooled transcriptome, and you would get most of the reads that were just mapping to rice genome, but there were certain reads that were left over. So we used those leftover reads to map on the fungal genome. So we could identify certain stage-specific proteins that are being expressed by this necrotrophic pathogen during its invasion in the cells. But there were certain proteins that were commonly expressed at all stages of infection. So we could identify around 790 genes which were expressed at all stages of infection. So this is a broader view of what we observed is, this is a heat map which tells us about the expression of genes. There is a scale of this heat map which tells like low expression or high expression. The red color, it shows high level of expression, and the green color, low expression. So as you can see from this heat map, out of these 790 genes, we could identify genes that were involved in cell wall remodeling. There were certain kinases which are important for signaling things. There were certain transporters. Different kind of genes were there, which is obvious because when pathogen has to infect inside the plant, it has to have a suite of different strategies to kill the cells. So, and also at the same point, it has to grow also. So we observed these kind of things. But what was most interesting for us is when we did the metabolic pathway analysis of this transcriptome data, we observed that there is a pathway which is called shikimate pathway, shikimic acid pathway. The genes related to this shikimate pathway they are all red. they are present in very high fold. Whereas there is another pathway on the left side, which is ergostol biosynthesis pathway. The genes were here, suppressed, but the shikimate pathway, this is not very visible, so this is a simplified version of this shikimate pathway. You see it starts by condensation of PEP and it follows 4-phosphate from 2 different pathways, and there are 7-8 genes which are leading to production of, different aromatic amino acids. So this shikimate pathway is important for production of amino acids. So if pathogen has to, progress, it has to, grow inside cells, it has to be metabolically very, very active. And for that, it would need proteins. It has to synthesize proteins. And these amino acids, probably they are helpful for the growth of this pathogen. So once we, identify this, that this pathway is important for, infection and proliferation of this pathogen inside the cells, we validated the expression of these genes using real-time PCR analysis, which is again corroborating with what we observed from the RNA-seq data. So question is, what next? Now we know that this shikimate pathway is important, but what next? So we discussed and I asked my students, like, what can we do? Can we block this pathway? So yes, because I was teaching this biotechnology courses at PAU Ludhiana, and I was quite familiar with this thing that I discussed earlier also, that this glyphosate is a known inhibitor of EPSP synthase. EPSP synthase is a rate limiting step here. Rate limiting step, one penultimate to the last reaction, EPSP synthase. It is a rate limiting step of EPS and this shikimate pathway. So, this thing was already known in the literature that glyphosate can inhibit the EPSP synthase of this EPSP synthase. People have exploited this strategy for some other purpose, but with regard to this fungicidal activity, this EPSP, the inhibition of EPSP synthase was not known. So what we did is we procured this glyphosate and treated this fungus with different concentrations of EPSP synthase, and what we can see is this glyphosate is inhibiting the fungus in a dose-dependent manner. So we also tested whether this glyphosate can inhibit other fungus also. So we also observed that other fungus are also, either they have reduced growth or they are being killed. So basically EPSP synthase is kind of a conserved, this shikimate pathway is a conserved pathway among all plants and animals. Plants also have these pathways. So, but, by means of this experiment, what we established is that if we can just block this pathway, we will be able to overcome this pathogen. So this was just as a proof of concept. But we cannot, recommend people to use glyphosate because it has been banned, in India because of some environmental concerns. Initially it was used, but nowadays it is not being used. So we did a few more experiments just to establish that this glyphosate treatment is, inhibiting this fungus to infect. So we again did some certain kind of experiments. We again showed that once you pre-treat this fungal sclerotia and then infect the plant, it is able to grow. But this is here, the question is, because now it is inhibiting the pathogen, how sure you are that it is directly targeting the EPSP synthase? There might be some alternate targets also. So one thing was to show that actually this protein, this glyphosate, is interacting with the EPSP synthase. What we did is we expressed this, we cloned this protein from fungus, expressed in E. coli, verified the protein, and using different interaction assays, we showed that this EPSP synthase is actually interacting with the glyphosate. Now, to just proceed further, because this was just a proof of concept, we wanted to proceed further, we wanted to exploit this information. So how can we exploit this information? So we looked at the structure of these proteins in plants versus pathogen. So we could see that there are some certain structural differences in plant-based EPSP synthase versus this naive EPSP synthase. There were certain residues which were distinct for binding of this glyphosate with the EPSP synthase of hydroquinone. So these were just structural differences which can be exploited to develop certain kind of inhibitors which are specific to rhizokinase solenoid. But what was more interesting for us, when we did comparative sequence analysis, we found a region in the fungal EPSP synthase which was very distinct. It was not present anywhere, not in any of the plant species. There was 200, 300, 300 base pair sequence which was exclusively present in the fungal EPSP synthase. So that was very exciting for us because I knew there is one technique that we can use to target now this distinct region. So there is a technique called host-induced gene silencing, where what we do is we develop double-stranded RNAs against that particular region. So what we did is we designed these double-stranded RNAs and we did fluorescein labeling of that double-stranded RNA and did incubation studies whether this fungal hypha can uptake these double-stranded RNAs inside them or not. So with this experiment we showed that if we deliver these double-stranded RNAs, the fungus can take them up. So this was exciting because then there is a mechanism inside the plants where these double-stranded RNAs are being converted by the host machinery, plant machinery, to convert them into small interfering RNAs. So they will be basically cleaved into small 21 nucleotide, small RNAs, which can target the fungal EPSP synthase. So what we did is we developed engineered one construct, and this is called host-induced gene silencing construct. Basically, we put these double-stranded RNAs in the construct, put inside them in agrobacterium, and infiltrated the Nicotiana. Nicotiana is a model plant where we can do certain studies without compromising time. So what we observed here is that when we have empty vector control, having no double-stranded RNA, the fungus is able to infect. So you see a lesion in the leaves. But when you silence this region, we have this Higgs construct, you see there is no development of fungal lesion. So this was very encouraging for us because we could show that just by targeting that unique region of fungal EPSP synthase, there was reduction in the fungal invasion. So after showing this, we did it in rice. So we developed stable transgenic lines, and you see compared to the control here, on the left side control, it is fully infected. We have 3 different rice transgenic lines, although there is certain level of infection, but it is very, very low. So we also wanted to see whether this, because we are putting extra pressure on the plant, we are giving extra system inside the plant which is producing double-stranded RNA, whether it is affecting the agronomic traits of rice or not. So we evaluated rice for different agronomic traits, and we didn't see any compromise in the agronomic traits. So this was this work we published in Plant Biotechnology recently in 2026. So this is all about this work. So in summary, this transcriptome analysis suggested an important role of genes involved in cell wall remodeling, toxin detoxification, transport, and transcriptional regulators in pathogen virulence, and the CQB pathway emerged as an important pathway in pathogen growth and virulence. This RS-EPSP has unique mechanism of inhibition by glyphosate, a step towards development of novel inhibitors, and finally inhibition of CQB pathway by host-induced silencing of EPSP impaired the virulence of also like to infect tobacco and rice. So this is all about my presentation. The idea was just to give you a message that enzymes are important and we can exploit them for different applications. And finally, this is my lab, my students who have done this work, and some of the funding agencies, CSIR, DBT, ICMR, so we have funding from different funding agencies. Thank you very much, and I will be happy to take a question.
Good afternoon, sir. Sir, my question is, what are the sources of SOD and what is its potential application?
What are different sources?
Of SOD?
Okay, so every aerobic organism, whether it be a plant or animal, all have SODs. सेल्स है ना, सेल्स में कंपार्टमेंटलाइजेशन है. क्लोरोप्लास्ट है, वहां पे अलग एसओडी होगा, माइटोकॉन्ड्रिया में डिफरेंट काइंड ऑफ एसओडी होगा, साइटोसोल में अलग एसओडी होगा. So based on the metal cofactor they use and based on which part of the cell they are divided into different categories. So these are called basically isocytosine, but the function is almost same. They are doing just dismutation of superoxide. So they are present in all aromas. But the expression of these genes it varies from individual to individual. और एप्लीकेशन क्या है इसका? एप्लीकेशन बेटे ऐसा है कि जैसे लेट्स से प्लांट्स की बात करें, वंस प्लांट्स आर अंडर स्ट्रेस कंडीशंस, बहुत ज्यादा स्ट्रेस जब होता है तो उसके फोटोसिंथेसिस इंपेयर होता है, रिएक्टिव ऑक्सीजन स्पीसीज बनती हैं, इलेक्ट्रॉन ट्रांसपोर्ट चेंज जब होता है तो उसमें से भी इलेक्ट्रॉन्स लीक होते हैं बहुत ज्यादा क्वांटिटी में एंड दे रिएक्ट विद दिस ऑक्सीजन से रिएक्ट करते हैं तो सुपरऑक्साइड आयंस बनते हैं एंड दे आर वेरी रिएक्टिव, बहुत ज्यादा फास्ट होते हैं वो, दे रिएक्ट विद एनी बायोलॉजिकल मैक्रोमोलिक्यूल। तो उससे क्या होता है कि वो म्यूटेशन हो जाती है सेल्स में, अगर लिपिड से काम करेंगे तो लिपिड पर ऑक्सीडेशन हो जाएगा और फिर क्या है कि बेसिकली सेल डैमेज हो जाता है। सो आई मीन इनको कंट्रोल करना बहुत जरूरी है। दे आर इंपॉर्टेंट इन वेरी लो क्वांटिटीज। जो रिएक्टिव ऑक्सीजन स्पीसीज बोले इनका कुछ सिग्नलिंग रोल्स भी होता है, जैसे H2O2 बोला मैंने कि सुपरऑक्साइड आयन बना, सुपरऑक्साइड आयन को कॉपर्जिंक एसओडी या MnSOD या कोई भी एसओडी है उसने उसको डिसम्यूटेट कर दिया इनटू H2O2। नाउ H2O2 इज ए सिग्नलिंग मॉलिक्यूल बिकॉज इट कैन डिफ्यूज। ये जो दूसरे वाले हैं सुपरऑक्साइड 9 सिंगलेट ऑक्सीजन दे कैन डिफ्यूज, उनको वहीं पे मारना पड़ेगा, अदरवाइज दे विल बी टॉक्सिक, बट H2O2 कैन डिफ्यूज। तो थोड़ा बहुत जो प्रोडक्शन है H2O2 का, बिकॉज देन वंस इट डिफ्यूजेस इट अल्टर्स न्यूक्लियर जीन एक्सप्रेशन। तो स्ट्रेस कंडीशन में क्या होता है, न्यूक्लियर जीन एक्सप्रेशन को ये अल्टर करता है, तो उससे प्लांट को अडेप्टेशन के लिए हेल्प मिलता है ना। तो देयर हैज टू बी फाइन बैलेंस बिटवीन प्रोडक्शन ऑफ दिस प्रोडक्शन एंड स्क्वेंजिंग ऑफ सुपरऑक्साइड एनाइंस। ठीक है, ऐसे ही ये माइक्रोब्स में भी मिलते हैं, है ना। लगभग जो जितने एरोबिक ऑर्गेनिजम्स हैं यू विल फाइंड एसओडीज, क्योंकि दीज आर ऑफ एंशिएंट ओरिजिन। बेसिकली क्या था कि जब लाइफ इवॉल्व हुई थी पहले तो ऑक्सीजन यूज नहीं होती थी, बट एज एवोल्यूशन हैपन ऑक्सीजन को यूज करना किया, एरोबिक ऑर्गेनिजम्स बने, एटीपी बन, ये सब यूज बनाने के लिए। तो उसी टाइम ही एवोल्यूशन के दौरान ये जो ये है ये माइक्रोबियल रीजन के हैं बेसिकली, जो एसओडीज वगैरह, क्योंकि पहले एनारोबिक एनवायरमेंट्स थी तो क्या ये एज ए सिम्बायोट्स दे एसोसिएटेड विद प्लांट्स, तो ये माइटोकॉन्ड्रिया क्लोरोप्लास्ट वगैरह जो है दे आर ऑफ एंशिएंट ओरिजिन, तो ट्रांसफर हुए हैं वैसे ही। और एवोल्यूशन हुआ है बिकॉज देयर वाज एवोल्यूशनरी प्रेशर, क्योंकि काफी सारे इवेंट्स ऐसे हुए जहां पे बहुत हार्श एनवायरनमेंट था, हार्श है ना, तो काफी रिड्यूसिंग एनवायरनमेंट था, फिर ऑक्सीडाइजिंग एनवायरनमेंट पे है, तो इन सब एंजाइम्स की जरूरत पड़ी। और एप्लीकेशन की बात करें तो एप्लीकेशन एसओडी की इंडस्ट्री में काफी सारी इंपॉर्टेंस है। इफ यू वांट लेट्स से इफ यू वांट टू इंजीनियर प्लांट्स फॉर एबायोटिक स्ट्रेस टॉलरेंट, तो वहां पे इंपॉर्टेंट रोल हो सकता है। इसमें एक जैसे फार्मास्यूटिकल इंडस्ट्रीज में भी इसका रोल है। आपको ये जो अपनी ग्राफ्ट एंड ट्रांसप्लांटेशन करते हैं, कुछ ऑर्गन्स प्रिजर्व करने होते हैं, कॉर्निया को जैसे वो करना है, तो यू कैन प्रिजर्व दिस इफ यू हैव दिस काइंड ऑफ एंजाइमेटिक सॉल्यूशन, तो प्रिजर्वेशन में हेल्प करते हैं। ठीक है, एनी अदर क्वेश्चन? Is there any strategy to reduce? Yes, but yes, there are a lot of strategies. तो बेसिकली मैंने पहले बताया कि जो ब्रीडर्स होते हैं और जो पैथोलॉजिस्ट होते हैं ये रिलाई करते हैं बेस्ड ऑन नेचुरल रेजिस्टेंस इन प्लांट्स, है ना। तो जैसे राइस की बात करें तो इसके बहुत सारे एक्सेशंस ऑलरेडी अवेलेबल हैं, वाइल्ड स्पीसीज बहुत सारी है, कुछ 1400-1500 इसकी वाइल्ड एक्सेशंस अवेलेबल हैं। तो लोग क्या करते हैं, जैसे लेट्स से एक डिजीज आ गया तो उसके अगेंस्ट उनको फिनोटाइप करते हैं। फिनोटाइपिंग मींस उनको चैलेंज करेंगे और उसको डिजीज स्कोरिंग देखते हैं एंड बेस्ड ऑन दैट दे आइडेंटिफाई कि कौन सा रेजिस्टेंट है, कौन सा ससेप्टिबल है। फिर क्या करेंगे बेसिकली कि जो रेजिस्टेंट होगा उसके लिए कुछ जीन्स रिस्पॉन्सिबल होंगे, है ना। तो क्लासिकल ब्रीडिंग वहां पे इंपॉर्टेंट रोल प्ले करती है। सो क्लासिकल ब्रीडिंग से क्या करते हैं बेसिकली दे क्रॉस इन द इंडिविजुअल्स, और उससे होगा क्या, देयर विल बी सर्टेन सेगमेंट्स ऑफ द जीन्स, क्योंकि मियोसिस होगा, क्रॉसिंग ओवर होगी, तो सर्टेन फ्रेगमेंट्स ऑफ द जीनोम दे विल बी ट्रांसफर्ड टू द ससेप्टिबल पेरेंट। तो उससे क्या होगा, जब वो फ्रेगमेंट उसमें आ जाएगा तो ससेप्टिबल वाला भी जो है दैट विल आल्सो बिकम टॉलरेंट। तो ये स्ट्रेटजी जो है ये करते हैं मार्कर्स। इसमें बेसिकली क्या है कि जो क्रोमोसोम वाइज लोगों ने कुछ मार्कर्स बना रखते हैं, मार्कर्स की फ्लैग मतलब जस्ट फ्लैग्स इन द क्रोमोसोम, कि ये फलानी जगह पे एक मार्कर मतलब सीक्वेंस है बेसिकली, वो देयर आर डिफरेंट काइंड ऑफ मार्कर्स। तो बेसिकली उसकी उन्होंने पूरे क्रोमोसोम को उन्होंने मार्किंग कर दी है और फिर मार्कर रेसिस्टेड सिलेक्शन करते हैं कि इस रीजन पे ये वाले मार्कर्स जो दे आर लिंक्ड मार्कर्स, तो जब क्रॉसिंग होगी तो ये उधर चले जाएंगे ट्रांसफर, तो दैट इज मार्कर रेसिस्टेड सिलेक्शन। दूसरा होगा बेटे ट्रांसजेनिक टेक्नोलॉजीज। ट्रांसजेनिक टेक्नोलॉजीज में क्या है, लेट्स से, क्योंकि ये जो पैथोजन इंटरेक्शन जो है दे आर डिफरेंट टाइप्स, बायोटिक इंटरेक्शन है, क्या बोलते हैं उसको, नेक्रोट्रॉपिक इंटरेक्शन है, हेमियोबायोट्रॉपिक इंटरेक्शन है ना, मतलब डिपेंड्स कि किस तरह की इंटरेक्शन है। कुछ ऐसी इंटरेक्शन होती हैं जहां पे जो हमारे प्लांट्स हैं उनमें कुछ मैकेनिज्म्स होते हैं विच कैन रिकॉग्नाइज द फंगल विरुलेंस जीन्स। तो उसमें क्या होता है कि यहाँ पे रिएक्शन एक रिस्पॉन्स आता है कि आपने पैथोजन को रिकॉग्नाइज कर लिया, प्लांट के पास एक जीन था उसने रिकॉग्नाइज किया, जो प्लांट ने उसने रिकॉग्नाइज किया, देयर इज वन टू वन रिएक्शन, दिस इज कॉल्ड, तो उसमें प्लांट में एक बड़ा रोबस्ट सा रिस्पॉन्स होता है, उसको बोलते हैं हाइपर सेंसिटिव रिस्पॉन्स। हाइपर सेंसिटिव रिस्पॉन्स में क्या होता है, प्रोग्राम्स एक सेल डेथ हो जाती है, उस लोकलाइज सेल डेथ होती है ना। तो बेसिकली प्लांट क्या करता है, अपने आप को मार देता है उस रीजन पे जहां पे इन्फेक्ट करने की कोशिश करता है ना। तो बायोट्रॉपिक इंटरेक्शन में नॉर्मली इस तरह की इंटरेक्शन होती है, क्योंकि अपनी अपनी स्ट्रेटजीज है, पैथोजन को भी सक्सेस उसमें इन्वेट करना है, होस्ट को अपने आप को अवॉइड करना है उससे। तो उस केस में क्या है कि वन टू वन इंटरेक्शन, अगर प्लांट को पता लगे कि ये इन्फेक्ट कर रहा है तो उसको पास और सेक्टर है, उसका बेसिकली मैकेनिज्म है ये, तो उसको वो पकड़ा गया तो अपने आप को किल कर लेगा वो, ठीक है। और भी बड़ी सारी स्ट्रेटजीज हैं। तो ट्रांसजेनिक टेक्नोलॉजी से आप जैसे आर जीन्स बोलते हैं, उनको रेजिस्टेंट जीन्स बोलते हैं, तो आप रेजिस्टेंट जीन्स को आप ओवर एक्सप्रेस कर दोगे तो प्लांट टॉलरेंट बन जाएगा। तो और बहुत सारी टूल्स एंड टेक्निक्स हैं जिनोम एडिटिंग से कर रहे हैं। मैंने जैसे बोला कि ससेप्टिबिलिटी इंड्यूस कर देते हैं, टॉक्सिन सिक्रीट करेंगे पैथोजेंस, या कुछ और तरीके से ससेप्टिबिलिटी इंड्यूस करेंगे, ये प्लांट की डिफेंस पाथवेज को हाईजैक कर लेते हैं, दे सिंपली इंटरफेयर विद द डिफेंस पाथवेज। तो आपको अगर पता चल जाए कि आपके जो डिफेंस पाथवेज में कहां जैसे ब्रेक हो रही है, पैथोजन कहां पे रिसीव कर रहा है, तो आप उस जगह पे मैनिपुलेट कर सकते हैं। यू कैन इवन डू प्रिसाइजली एडिट दैट पर्टिकुलर जीन, ठीक है, उसके एक्सप्रेशन को चेंज करके मॉड्यूलेट करके थोड़ा सा। तो देयर आर एन नंबर ऑफ स्ट्रेटजीज। दूसरा मैंने आपको बताया कि सिंपली केमिकल टेक्नोलॉजीज, क्या आपको पता है कि ये फलाना जो एंजाइम है पैथोजन का इंपॉर्टेंट है, यू डिजाइन सम सर्टेन केमिकल पेस्टिसाइड अगेंस्ट दैट, जो केमिकल जो उसको उसके एंजाइम को इनहिबिट कर दे। So these are different strategies. Any other question? If not, thank you.
Thank you, Dr. Arun Kumar, for such a informative and interactive session. Thank you so much.